Genetic testing is not the finish line. In inborn errors of metabolism, or IEMs, it is more like a molecular detective story. Many parents believe that once a child has a blood draw and one genetic test is done, the answer will be clear. Does my child have a genetic disease or not? But in real life, it is often not that simple. This is especially true for IEMs, inborn errors of metabolism. A genetic report can be very important, but it is not an all-powerful judge. A negative result does not mean the case is closed, and a positive result does not automatically prove the whole diagnosis. A real diagnosis has to look at the child's symptoms, the family history, metabolic testing, enzyme testing, imaging studies, and how the illness has unfolded over time. All of these pieces of evidence have to be put together before the true picture can begin to emerge. When many children first arrive in the emergency room, their parents usually cannot name a specific disease. They can only describe scattered pieces of the story. Every time this child has a fever, he becomes so sleepy that he is hard to wake. If she eats just a little less, she starts vomiting. For another child, what looks like an ordinary cold is followed by sudden seizures. At times, it may look like a psychiatric problem. At times, it may look like developmental regression. Sometimes it looks like epilepsy. Sometimes it looks like encephalitis. In the beginning, these children may be placed under many different diagnoses: epilepsy, encephalitis, cerebral palsy, autism spectrum disorder, behavioral or psychiatric problems. And the stack of test results keeps getting thicker. EEG, brain MRI, complete blood count, liver and kidney function tests, blood ammonia, lactate, blood gas, plasma amino acids, urine organic acids, acylcarnitine profile. Every report is like one piece of a puzzle. The real turning point often comes when one group of results suddenly begins to fit. For example, a child has repeated vomiting, marked sleepiness, changes in consciousness, elevated blood ammonia, an abnormal amino acid profile, and a urine organic acid test showing certain metabolites that are clearly elevated. That is when doctors begin to ask a different question. Could this be more than a primary neurologic disorder? Could it be a urea cycle disorder, an organic acidemia, a fatty acid oxidation disorder, a mitochondrial disease, or another type of IEM? This is when genetic testing truly enters the investigation. It is like a molecular fingerprint at the crime scene. It can help doctors confirm a molecular diagnosis. It can help identify the specific disease subtype. It can help assess carrier risk in relatives. And it may affect treatment choices, dietary management, family planning, prenatal diagnosis, or pre-implantation genetic testing. But remember this: if a child is actively seizing, extremely sleepy, confused or unconscious, or vomiting again and again, if the blood ammonia is very high, if the blood sugar is abnormal, or if the blood gas already looks dangerous, doctors will not wait for the genetic report before they begin treatment. In a metabolic crisis, time is brain cells. Especially with hyperammonemia, hypoglycemia, or severe acidosis, delayed treatment can cause irreversible brain injury. So, without delaying treatment, doctors try to collect the most important samples as quickly as possible: blood, urine, and, when needed, cerebrospinal fluid or a DNA sample. Because once treatment begins, many metabolites may start returning toward normal. If that diagnostic window is missed, it can become much harder to capture the evidence later. The diagnosis of an IEM is usually not a straight line. It is more like several lines of work moving forward at the same time. In the emergency room, the first goal is to stabilize the child's vital signs. At the same time, doctors try to save key metabolic samples. Metabolic testing looks for traces at the scene. Genetic testing looks for the molecular cause. Enzyme testing, RNA analysis, mitochondrial DNA testing, CNV analysis, and functional studies can all help fill in the missing evidence. If the child is critically ill, if the phenotype is complex, or if several organ systems are involved, rapid whole exome sequencing or rapid whole genome sequencing may be done at the same time as metabolic treatment. Genetic testing is like tracking the suspect afterward. Emergency treatment is like putting out the fire first. When the fire is still burning, you cannot wait for the detective report. Chapter 1. What is genetic testing actually looking for? Many parents think genetic testing simply means drawing a tube of blood and checking whether the child has a genetic disease. But it is much more complex than that. Testing related to genetic diagnosis may include several categories: genetic testing, cytogenetic testing, metabolic biochemical testing, enzyme assays, and functional studies. Genetic testing mainly looks for several kinds of clues. The first kind is a change in the DNA sequence. For example, one base in a gene may be changed, or a small piece may be missing, or a small piece may be added. These changes can cause an enzyme to fail. They can cause a transporter to fail. They can disrupt a cofactor pathway. They can also affect how an organelle functions. Many IEMs happen in exactly this way. A metabolic pathway is like a production line. If one worker on that line suddenly stops working, raw materials begin to pile up. The final product may not be made in enough quantity, and inside the cell, the production line starts to jam. The second kind of clue is a larger gain or loss of DNA. These changes are often called CNVs, or copy number variants. In simple terms, one part of a gene may be missing a copy, may have an extra copy, or a chromosome segment may have a structural change. Standard sequencing does not always see these problems clearly. For example, some large deletions, duplications, complex structural variants, repeat expansions, pseudogene interference, or low-level mosaicism may be missed by routine methods. These situations may require specialized testing. For example, deletion and duplication analysis, chromosomal microarray, MLPA, FISH, or other techniques. The third kind of clue is at the RNA level. DNA is like the blueprint stored in the warehouse. RNA is like the work order copied from that blueprint. Sometimes the blueprint does not appear to have an obvious problem, but the work order has been cut and assembled incorrectly. One section may be missing, one section may be added, or the pieces may be spliced together in the wrong order. RNA analysis can help doctors determine whether certain splicing abnormalities, deep intronic variants, or abnormal expression patterns are actually affecting gene function. But a negative RNA result in blood does not mean there is no problem in the liver, the brain, or the muscle. The fourth kind of evidence comes from enzyme testing and functional studies. At the core of many IEMs, the key question is not simply whether something is written on a genetic report. The real question is whether the machinery inside the cell is actually working. Is the enzyme working? Is the transporter working? Is the cofactor pathway working? Is the mitochondrion, the cell's power plant, losing power? That is why, in some cases, enzyme testing and cellular functional studies may bring doctors closer to the actual disease process than the DNA report alone. If a child's symptoms fit, the metabolic markers fit, and the enzyme activity is clearly reduced, then even if genetic testing has not yet found the complete answer, we should not casually conclude that the disease is not there. Chapter 2. Common types of genetic testing. Different tools see different kinds of evidence. The first type is single-site testing. This test usually looks at one known position in the DNA. For example, if a specific variant has already been found in the family, then relatives may only need to be tested for that one site. It is like taking a suspect's fingerprint and comparing it with evidence from the scene. It is fast, it costs less, and the target is very clear. But its limitations are just as clear. It only looks at one location. If the problem is somewhere else, this test will not see it. The second type is single gene testing. This test usually looks at one gene. When a child's features are very typical, and the doctor strongly suspects a disease linked to one specific gene, single gene testing can be very efficient. But IEMs can be difficult. Many inborn errors of metabolism have overlapping symptoms: vomiting, seizures, developmental delay, psychiatric-like symptoms, liver injury, muscle disease. The same symptoms can come from completely different metabolic pathways, so if single gene testing is aimed in the wrong direction, the test may miss the real disease. But when the phenotype is highly specific, single gene testing can still be a very useful tool. The third type is a targeted gene panel. This means testing a group of genes related to one category of disease. For example, a urea cycle disorder panel, an organic acidemia panel, a fatty acid oxidation disorder panel, a mitochondrial disease panel, or a panel for epilepsy and developmental delay. It is like the doctor has already narrowed the search to a few neighborhoods, and now the detectives focus their search there. The advantage of a panel is that the cost is usually more manageable, the results are more focused, the depth of coverage is often better, and there are usually fewer incidental findings than with whole exome or whole genome sequencing. When the child's biochemical results or clinical features already point toward a certain group of metabolic disorders, a panel can be very practical. But panels also have blind spots. If a gene is not included in the panel, the test cannot find it. If a newly discovered disease gene has not yet been added, the panel will not detect it either. Deep intronic variants, regulatory regions, complex structural variants, and some deletions or duplications may also be missed. So a negative panel result only means this: within the scope of that panel, there was not enough evidence to explain the child's condition. It does not prove that the child does not have an IEM. There is another point parents should understand. Some panels include candidate genes with limited evidence linking them to disease. That can increase the chance of finding a VUS, a variant of uncertain significance. So when choosing a panel, the question is not only how many genes are included. The more important question is how strong the evidence is between those genes and the diseases being tested. The fourth type is whole exome sequencing, or WES. Exons are the parts of genes that code for proteins. The goal of WES is to capture most known exons across the genome, but WES does not reliably cover every exon, every transcript, or every hard-to-sequence region. Protein coding regions make up only a small part of the human genome, roughly around 2%. But many known disease-causing variants are found in these regions. That is why WES is widely used in children with developmental delay, intellectual disability, epilepsy, congenital anomalies, and difficult-to-diagnose genetic conditions. It is like searching first in the parts of the city where the clues are most likely to be hidden. The advantage of WES is that it can look at many genes at once. Some laboratories can also perform a more open analysis, which may identify genes that were not included on a targeted panel. For children with complex features or no clear diagnostic direction, WES can be very valuable. But the limitations of WES are also very important. WES mainly looks at exons. It has limited ability to detect problems in deep intronic regions, promoters, enhancers, regulatory regions, non-coding regions, some CNVs, large structural variants, repeat expansions, complex rearrangements, pseudogene regions, and mitochondrial DNA. So sometimes a situation like this happens. The child's symptoms and metabolic pattern look very much like an autosomal recessive disorder, but the report finds only one pathogenic variant. At that point, we cannot immediately say, this is not the disease. Doctors have to keep asking questions. Could the second variant be hidden in a place that WES cannot see well? Is there a deletion or duplication? Is there a splicing abnormality? Is there a deep intronic variant? Is there a coverage problem? Did the analysis pipeline fail to capture something? This is one of the places where the molecular detective story most often gets stuck. The fifth type is whole genome sequencing, or WGS. WGS has a broader search range. It looks not only at exons, but also at introns, intergenic regions, and some regulatory regions. For certain structural variants, non-coding variants, and copy number variants, WGS may have advantages over WES. If WES or a panel is negative, but the doctor still strongly suspects a genetic disorder, WGS may be the next step. But WGS is not a magic eye. Sequencing more DNA does not mean that every finding can be explained. The amount of data is huge. The cost is higher. The analysis is more complex. And there may be more variants of uncertain significance. Complex repeats, low-level mosaicism, and certain structural variants can still be missed. Methylation abnormalities and imprinting disorders usually require specialized testing. So the biggest challenge with WGS is not always that it sees too little. Sometimes it sees so much that the real question becomes, which finding is the true culprit? The sixth type is mitochondrial genome testing. Do not forget, there is another power plant inside the cell. It is called the mitochondrion. Mitochondrial disease is especially easy to misdiagnose. It can affect many organ systems, and the symptoms can vary widely. Some children may have seizures, developmental regression, muscle weakness, external ophthalmoplegia, hearing problems, cardiomyopathy, elevated lactate, or abnormal brain imaging. Other patients may have diabetes, optic atrophy, kidney tubule problems, gastrointestinal dysmotility, migraine-like episodes, stroke-like episodes, or even psychiatric or behavioral changes. Some patients may seem only mildly affected most of the time. They may be easily fatigued. They may have exercise intolerance. Then an infection comes along, and they suddenly get much worse. Mitochondrial diseases can involve two sets of genetic clues. One set is in the nuclear DNA. The other set is in the mitochondrial DNA. A standard WES test is not the same thing as complete mitochondrial testing. Mitochondrial DNA also has a special issue called heteroplasmy. In simple terms, the percentage of a mitochondrial variant can differ from one tissue to another in the same person. A normal-looking blood result does not mean the muscle is normal. It does not mean the brain tissue is normal. It does not mean the kidney tubule cells are normal. And it does not mean the urinary sediment cells are normal. So when mitochondrial disease is suspected, the type of sample itself becomes part of the diagnostic strategy. Doctors may consider mitochondrial genome sequencing, a nuclear gene panel, WES, WGS, and when needed, testing urinary sediment cells, a muscle sample, respiratory chain enzyme studies, muscle pathology, or cellular functional studies. The seventh type is RNA sequencing and functional testing. These tools are often used when the DNA report does not fully explain the case. Some variants are difficult to interpret. A variant may be hidden in an intron. It may affect splicing. It may cause a gene to be expressed at a very low level, or it may damage the RNA, the work order copied from the DNA blueprint. RNA-EQ can help answer several questions. Is there abnormal splicing? Is an exon being skipped? Is there an abnormal transcript? Is there intron retention? Is this gene being expressed at a clearly reduced level? In difficult diagnostic cases, RNA-seq can be used as a follow-up test after negative WES or WGS, but it also has limitations. Different tissues express different genes. What can be seen in blood may not be seen in the brain, the liver, or the muscle. Many genes related to metabolic disorders are most active in specific tissues, and those tissues may not be easy to sample. So a negative RNA result in blood does not rule out an IEM. Some genes are not expressed in blood. Some are expressed at very low levels, and some abnormal RNA molecules may be destroyed by the cell before they can be detected. So to summarize, if doctors already strongly suspect a certain category of IEM, they may consider a targeted panel, single-gene testing, or single-site testing. If the child has a complex phenotype, developmental delay, intellectual disability, epilepsy, or involvement of multiple organ systems, WES or WGS may enter the diagnostic process earlier. Sometimes doing it earlier is more effective than moving step by step through many separate tests. If mitochondrial disease is suspected, always remember there are two genetic lines to consider. One is mitochondrial DNA, the other is nuclear DNA. Genetic testing is powerful, but it is not the finish line. In IEMs, what can truly help a child is not staring at one report and making a final judgment. It is putting together the symptoms, the metabolic clues, the genetic evidence, the enzyme results, and the functional studies piece by piece, until all the evidence points in the same direction. Chapter 3. The 5 types of results on a genetic report. Results on a genetic report are usually classified into several categories. Pathogenic, likely pathogenic, variant of uncertain significance, likely benign, benign. The result that often causes the most anxiety for parents is the variant of uncertain significance, or VUS. A VUS is not a diagnosis. It is also not a rule-out. It is more like a blurry fingerprint found at the scene. It is worth saving. It is worth rechecking. But it cannot close the case by itself. A VUS means that there is not enough evidence right now. We cannot say that it definitely causes disease, and we cannot say that it definitely does not matter. That is why a VUS should not be used alone to decide treatment. It should not be used alone for prenatal diagnosis. It should not be used alone for pre-implantation genetic testing. And it should not be used to label relatives as affected. But there is one important point. If a child's symptoms strongly resemble a treatable IEM, and the metabolic testing also strongly supports it, doctors will not simply wait because the report says there is only a VUS. The clinical problem still has to be managed, because in IEMs, sometimes there is no time to lose. At that point, the goal is not to panic over one VUS. The goal is to build the evidence. Do the child's symptoms fit? Do the metabolic markers support it? Is the enzyme activity reduced? Are both parents carriers? Do the two variants come from opposite sides of the family? Are there other affected relatives? Is there new evidence in the medical literature or in genetic databases? The genetic report is only one piece of evidence. The real diagnosis comes from the doctor putting together the symptoms, the physical findings, the biochemical tests, the imaging studies, the family history, the genetic results, and the functional evidence. Chapter 4. Why a negative genetic test does not rule out an IEM. What worries doctors most is not that a report says negative. It is when families read negative and understand it as, "My child does not have the disease." Those are not the same thing. A negative genetic test only means this. Within the parts of the genome this test was able to see, it did not find a disease-causing variant strong enough to explain the child's symptoms, but problems can still be hidden in places the test cannot see. Take WES, whole exome sequencing, as an example. WES mainly looks at exons. If the problem is hidden in a deep intronic region, a promoter, an enhancer, or another regulatory region, WES may not detect it. If the problem is a large deletion, a duplication, an inversion, a complex rearrangement, or a repeat expansion, standard short-read sequencing may not see it clearly. Some genes also sit next to highly similar pseudogenes. It is like two streets that look almost identical, with house numbers that are easy to confuse. Sometimes sequencing can take the wrong road. There is also low-level mosaicism. This means the variant is present in only some cells. If the level is very low in blood, routine testing may miss it. Mitochondrial disease is even more complicated. The percentage of a mitochondrial DNA variant can differ from tissue to tissue. A negative blood test does not mean the muscle is negative. It does not mean the brain tissue is negative. And it does not mean renal tubular cells are negative. There is another possibility. Medicine may not yet know that this gene causes disease. A few years later, a new disease gene may be discovered. When the old report is reanalyzed, a clue may suddenly appear. So a negative genetic test is not a closing statement. It is more like a progress report in an ongoing investigation. For difficult IEM cases, we cannot close the case based on one test, one report, and one negative result. Chapter 5. The common dilemma in autosomal recessive disorders. Finding only one variant. The child's symptoms seem to fit. The metabolic markers also seem to fit. But the genetic report finds only one pathogenic variant. At that point, we should not simply say, "The child is only a carrier." Many IEMs are inherited in an autosomal recessive pattern. Usually, the child has to inherit one abnormal copy from each parent in order to develop the disease. But the second variant is not always located where routine testing can easily find it. It may be a large deletion. It may be a duplication. It may be hidden in a deep intronic region. It may affect splicing. It may be located in a promoter or regulatory region. It may be obscured by a pseudogene. It may be present as low-level mosaicism. Or it may simply not have been analyzed by the report at that time. So, if the biochemical evidence is strong, the next step is not always to immediately repeat the blood draw at another lab. Often, the more important step is to go back and review the original report. What exactly did it test? Did it include CNV analysis? Did it include deletion and duplication analysis? Did it include mitochondrial DNA? Were there any regions with low coverage? Should the child have additional RNA analysis, WGS, enzyme testing, or functional studies? In IEM diagnosis, biochemical evidence is extremely important. If the child has a typical metabolic crisis, specific abnormal metabolites, and clearly reduced enzyme activity, then even if the genetic answer is not complete yet, the clinical evidence should not be thrown away. A disease does not disappear just because a report says, "No clearly pathogenic variant identified." Chapter 6. A positive genetic test still has to fit the patient. When a report says pathogenic or likely pathogenic, we still cannot automatically attribute every symptom to that gene. Every person carries many genetic variants. Most are simply background variation. Some represent carrier status only. Some may have incomplete penetrance. Some may be linked to later onset disease. Some do not match the child's current symptoms at all, and some are incidental findings. So, after a positive genetic result, doctors still have to keep asking questions. Does this gene fit the child's presentation? Does the inheritance pattern fit? Are there enough variants to explain the disease? Do parental test results support the interpretation? Do the metabolic markers support it? Do the enzyme results support it? Is there another disease that explains the child's condition more convincingly? In IEMs, the most reliable diagnosis does not come from one report alone. It comes from multiple lines of evidence locking together. The clinical picture fits, the metabolic pattern fits, the genetic result fits, the enzyme testing or functional studies also fit. That is what a complete chain of evidence looks like. Chapter 7. When should further testing be considered? If a child is strongly suspected of having an IEM, but a panel or WES is negative, families can discuss next steps with a genetics specialist or metabolic disease specialist. First, review the original report again. Look at the scope of testing. Did it include CNV analysis? Did it include deletion and duplication analysis? Did it include mitochondrial DNA? Did it mention any regions with low coverage? Second, consider trio analysis with the child and both parents. This can help determine whether a variant came from the father or the mother, whether it is de novo, and whether two variants are on different copies of the gene. Third, in certain special situations, doctors may consider testing for uniparental disomy, regions of homozygosity, SNP array findings, or WGS-based analysis. For example, the child may appear to have a homozygous variant, but only one parent is a carrier, or there may be large regions of homozygosity or loss of heterozygosity, or the phenotype may suggest an imprinting disorder, or the genomic pattern may suggest parental relatedness. Fourth, add deletion and duplication analysis. This is especially important in recessive disorders when only one variant has been found. Fifth, consider moving from WES to GS. If doctors suspect that the problem is hidden in a deep intronic region, a regulatory region, or a structural variant, WGS may be more appropriate. Sixth, consider RNA analysis. When doctors suspect a splicing abnormality, abnormal gene expression, or an intronic variant affecting transcription, RNA analysis can help provide additional evidence. Seventh, consider mitochondrial genome testing. This is especially important when the child has elevated lactate, muscle disease, seizures, encephalopathy, external ophthalmoplegia, cardiomyopathy, or involvement of multiple organ systems. Eighth, consider enzyme testing, metabolic flux studies, or cellular functional studies. For some metabolic disorders, these tests come closer to the real disease mechanism than DNA testing alone. And one more point is very important. Do not casually discard the raw data. Genetic databases are updated. Disease genes are updated. Variant classifications are updated. Analysis software is updated. A negative report from today should not be locked away forever. Chapter 8. Before genetic testing, parents should ask the right questions. Before genetic testing is done, parents should try to understand what kind of test is actually being ordered. Is this single gene testing? A gene panel? Whole exome sequencing, or WES, whole genome sequencing, or WGS, or mitochondrial DNA testing? Does the test include CNV analysis? Can it detect exon-level deletions and duplications? Can it identify structural variants? Does it include mitochondrial DNA? What is the lower detection limit for heteroplasmy? Will the report mention regions with low coverage? Should the child and both parents have trio testing? Will the report include secondary findings? Is genetic counseling available? Can the raw data be stored? Can the data be reanalyzed in the future? If a VUS is found, can family segregation testing be done? Could the result affect future family planning, prenatal diagnosis, or PGTM, pre-implantation genetic testing for monogenic disorders? These questions are not nitpicking. They are part of protecting a child's chance at the right diagnosis. Chapter 9. The future of IEM diagnosis will not depend on one test alone. The future of diagnosing inborn errors of metabolism will look more and more like a careful review of many kinds of evidence. WGES can widen the search area. Long-read sequencing can help detect complex structural variants. RNA-seq can help explain abnormal splicing and abnormal gene expression. Metabolomics can search for abnormal metabolites. Proteomics and enzyme assays can help show the functional consequences. Artificial intelligence and phenotype databases may help doctors reorder candidate genes, and periodic reanalysis can allow old data to shine again under new medical knowledge. But no matter how far technology advances, the true starting point is always the patient. When did the child first become sick? What triggered the episode? Does fever make the child worse? Does fasting make the child worse? Does a high-protein meal make the child worse? Does exercise or infection cause sudden deterioration? Has there been vomiting, severe sleepiness, seizures, psychiatric-like symptoms, liver injury, muscle disease, cardiomyopathy, eye problems, hearing problems, skin or hair abnormalities? Is the blood ammonia high? Is the lactate high? Is the blood sugar abnormal? Is the acid-base balance disturbed? Are there specific abnormalities in amino acids, acylcarnitines, or urine organic acids? In the family, has there been sudden infant death, repeated miscarriages, death of male newborns, unexplained neonatal encephalopathy, parental relatedness, developmental delay, epilepsy, psychosis-like symptoms, unexplained liver failure, HELLP syndrome during pregnancy, or acute fatty liver of pregnancy? These clinical clues are the map for the genetic data. Without that map, even the most powerful sequencing test can get lost in a sea of data. Final message. So in the end, please remember this. For a child suspected of having an inborn error of metabolism, genetic testing is important, but it is not a standalone judge. In an acute metabolic crisis, doctors first have to treat hyperammonemia, hypoglycemia, acidosis, seizures, and altered consciousness. At the same time, without delaying treatment, they try to collect the key metabolic samples as quickly as possible. If the genetic result is positive, doctors still have to ask whether the classification, the inheritance pattern, the phenotype, the biochemical evidence, the enzyme results, and the functional evidence truly fit together. If the genetic result is negative, that still does not automatically rule out an IEM. The testing scope, structural variants, deep intronic variants, pseudogenes, low-level mosaicism, mitochondrial heteroplasmy, tissue-specific gene expression, and updates in medical knowledge can all leave blind spots. Finding only one pathogenic variant in a suspected recessive disorder should not end the investigation too quickly. A VUS is not a diagnosis. It should not be used alone to guide treatment. It should not be used alone to predict risk in relatives. It should not be used alone for prenatal diagnosis. And it should not be used alone for PGTM. It needs family testing, functional evidence, updated literature, and reanalysis over time. Diagnosing an IEM is like a medical detective story. Genetic testing provides a critical fingerprint. Metabolic testing provides traces from the scene. Enzyme testing and functional studies tell us whether the machinery inside the cell has actually stopped working. The clinical presentation and family history tell doctors which road to investigate. For example, hearing loss, diabetes, migraine-like episodes, cardiomyopathy, and stroke-like episodes, especially when they cluster along the maternal side of the family, should remind doctors to think about mitochondrial disease. A truly careful diagnosis does not come from staring at one report and making a final judgment. It comes from letting each piece of evidence check the others until they all point to the same cause.
If you are interested in inborn errors of metabolism, or IEMs, and in the connection between neurologic or psychiatric symptoms and inherited metabolic encephalopathies, you can find the full written version in the Shunyi series.